Welcome back to our channel. Today we are here to discuss important MCQs on tissue culture part 2. Coming to the question number 1. In plant tissue culture, the callus tissue are generated into a complete plantlet by altering the concentration of option A sugars, option B hormones, option C amino acids, option D vitamins and minerals. Here correct answer is hormones. By altering the concentration of hormones, it includes auxin, cytokinin, gibberellins, ethylene, and abscisic acid. Here, by altering the auxins and cytokinins for regeneration of shoot and root, and for equal amount of auxins and cytokinins results in the callus formation, and increase in the auxin concentrations results in the root development, uh, and increase in the cytokinin concentration results in the shoot development. Likewise, altering the concentration of hormones develop into the complete plantlet. Coming to the question number 2. What is the significance of aseptic technique in plant tissue culture? Option A. Prevent contamination of plant cell. Option B. Enhance nutrient absorption. Option C. Improve photosynthesis efficiency. Option D. Accelerate root development. In this aseptic technique means prevention of the contamination of any viral something pathogen, diseases, fungal, bacteria and everything. Prevents the contamination of the plant cells. Coming to the question number 3. Which of the following is widely used in the crop improvement and crop management? Here option A is the agrobacterium, B is the plasmid, option C is the cosmid, option D is the plasmid. Here widely used organism for crop improvement and crop management, that is the agrobacterium. With this agrobacterium we can introduce the origin of interest to control the any pest and diseases or improvement of the yield using the agrobacterium. This agrobacterium used here as the transporter or vector that whatever our gene interest is carried forward to the into the plant cell through agrobacterium, and these all are the vectors, as we know already discussed in the earlier plasmid, cosmid, plasmids. All these are vectors that are incorporated, we can incorporate it into the agrobacterium cells. And coming to the question number 4, which of the following growth hormones produces apical dominance? Option A ethylene, option B cytokinin, option C auxin, option D gibberellins. And here correct answer is auxins. Here production of apical dominance by auxins. Coming to the question number 5, hybrids are produced by option A the nucleus of one species but cytoplasm from the both parent species. Option B, the fusion of two same nuclei from the same species. Option C, the fusion of two different nuclei from the different species. Option D, none of the above. Here, hybrids means nucleus of one species along, but here this is the nucleus of one species, and here cytoplasm of the both species, parent species combines this hybrid production. And here correct answer is option A. Coming to the question number 6, which of the following chemicals are most widely used for protoplast fusion? Here option A mannitol, option B polyethylene glycol, option C sorbitol, option D mannose. And here for protoplast fusion we'll use polyethylene glycol, that is the we'll call it as the PEG. So here correct answer is option B. Question number 7. Who is the known as the father of tissue culture? Option A Bonner, option B Liebig, option D Haberland, option D Cothernett. And here correct answer is Haberland. Here father of tissue culture is Haberland. Question number 8. What is callus? Option A tissues that goes to form an embryo. B. An organized activity dividing the mass of cells maintained in a culture. Option C. An insoluble carbohydrate. Option D. A tissue that grows from an embryo. Here the correct answer is callus is an unorganized activity dividing the mass of cells maintained in the culture. We call it as callus. Question number 9. Which of the following is a key advantage of using micropropagation in plant tissue culture? Option A, genetic diversity. Option B, rapid clonal propagation. Option C, resistance to diseases. Option D, increased nutrient uptake. Here, advantage of micropropagation is a rapid clonal propagation, and this micropropagation regeneration plants throughout the year and the life cycle is so reduction. So using this micropropagation, we can produce lot of the propagules from here. Coming to the question number 10, which term describes the process of introducing foreign genes into plant cell using tissue culture? Option A, grafting. Option B, micropropagation. Option C, somatic hybridization. Option G, genetic engineering. Here, any introducing foreign genes into the plant cell, we can through development through this genetic engineering or DNA technology or recombinant DNA technology. Using this technique, we can introducing foreign genes into our plant based on our need. If we want to develop any resistant gene, we are going to take from the other organism and incorporate it into the plant cell and reproduce to the from reproduce to plant. That plant should maintain and resistance to a particular disease. That will the through genetic engineering. Coming to the question number 11, which type of explant is commonly used to insert tissue culture? Option A, seeds. Option B, roots. Option C, leaves. Option D, flowers. And here, commonly used explant, that is a seeds. Coming to the question number 12, what is the callus in the context of plant tissue culture? Option A specialized root structure. Option B, a mass of undifferentiated cell. Option C, a fungal infection in plant tissue. Option D, a disease-resistant plant variety. Here, callus means the mass of undifferentiated cell.